Great, and so I'll try to catch us up on time to tail end, so we'll have a good discussion. I'm Sandip Patel, I'm a medical oncologist at UC San Diego, where I focus on early phase immuno-oncologic development. These are my disclosures. I won't be discussing any off-label use in my presentation. And so really how I want to connect with the other presentations from earlier and also earlier today is the fundamental problem in cancer immunotherapy. Now, in liquid tumors, you have cells, you engineer them, and they can find the target. And depending on the type of liquid tumor, the importance of the niche and how immunosuppressive the niche is may or may not limit efficacy. In solid tumors, the niche is actually the most important in terms of limiting the ability of either existing or exogenous immune cells from attacking the tumor. And so this is essentially what a solid tumor immune microenvironment is. And to really hone in on the interaction that's gained the most traction in solid tumors, this is the interaction between the tumor cell and the cytotoxic T lymphocyte at the PD1 PD-L1 junction. And so some of the data I'm going to show in terms of the biomarker science really focuses on this junction in particular. But there are multiple synapses of clinical significance throughout the tumor microenvironment, and I want to focus on some of these. The first is a junction we often don't see in the solid tumor space, which often takes place in the tumor-draining lymph nodes, and that's the interaction between dendritic cells and the cytotoxic T lymphocyte. One of these interactions is the PD-L1 interaction, and then there are a myriad of other cell types that can be affected and that are targets of a current cell-based or cell-based therapies or immune checkpoint blockade, looking at TGF-beta blockade in terms of regulatory T-cell induction, Th2 T-cells, which are generated by a cocktail of cytokines, M2 macrophages, which are often stimulated by a CSF1 transition, which can now be inhibited, tumor-associated fibroblasts, which are a major issue in certain epithelial malignancies, such as pancreatic cancer, actually have an immunologic function as well. And one of the CAR constructs that Dan was mentioning with anti-mesothelin, the ability to overcome these tumor-associated fibroblasts within this hostile tumor microenvironment is a major issue in terms of utilizing these agents in solid tumors. And so the question of the cells and having them be generated to fight the right target is a truly important question and central in terms of anti-cancer efficacy. But making sure these cells are successful within a very hostile tumor microenvironment is really what I want to focus on in terms of giving these cells the additional tools they need in order to be successful in this hostile microenvironment. And so this is a cartoon that shows the interaction between tumor and T cell, or it could be an antigen-presenting cell. And I show the T cell just because these checkpoints are better understood. But essentially, you have the lock-in key, the first signal between the MHC peptide and the TCR, and the variety of checkpoints here in red, for which you have inhibitory antibodies, the most famous being the anti-PD1 PD-L1 agents. They're antibodies that block CTLA-4. And currently in clinic, we have a variety of novel checkpoints being investigated, LAG3, TIM3 on the T cell side, as well as TNF superreceptor family agonists. And so if you have an initial interaction, which you have a neoantigen from the cancer that unlocks a T cell, inhibiting a break is important, but if you can accelerate an accelerator, you can often augment an immunologic response. And most importantly, perhaps engender an effector memory T cell population to keep the cancer at bay for a longer period of time. And these are checkpoints such as OX40, 4-1BB, which Dan mentioned is in line in terms of the CAR T cell construct, and I'll talk a bit about this concept later on. And then if I were to show a different diagram with the NK cell, I'd be talking about inhibiting the KIR interaction, or perhaps having NKG2D ligands as activators of the NK cell. And so where do cellular therapies and immune checkpoint blockade, how do they play out in terms of the solid tumor landscape? And so each patient likely has a unique deficit in their tumor immune microenvironment. And so you have patients who have complete immunologic ignorance of their tumor. Here they have no PD-L1 expression or no T cells within the tumor microenvironment, or NK cells or any anti-cancer cells for that matter. And this is the immune desert, or total immunologic ignorance. You have patients who have PD-L1 upregulation and PD-L1 expression right at the border of the tumor, but they can't enter into the hostile tumor microenvironment, the T cells, to kill the tumor. And this is what's called adaptive resistance. Then you have patients in which you have tumor-infiltrating lymphocytes, you have effector T cells, but you don't have any PD-L1 expression, so these T cells are tolerant of the tumor around them. And these were those other checkpoints that I showed you in red and green on the previous slide that likely need to be unveiled, likely in a combinatorial manner to have these T cells recognize and attack the tumor. And then finally, kinase induction, where you have both where you have PD-L1 expression, but you have no T cells. And so there are various mechanisms in terms of upregulating STAT and ALK pathway signaling signaling that can result in PD-L1 upregulation. From an oncogenic standpoint, this is different than the adaptive PD-L1 expression we see that's immune-mediated. And depending on which patient you are, your solution in terms of fighting your cancer is going to be slightly different, and your therapeutic strategy differs. And so the majority of the sessions today have covered cellular therapeutics, and cellular therapeutics, which are actually probably the prime strategy for targeting patients who have a tumor that's essentially an immunologic desert. And so these are vaccination strategies, cell-based therapies in which you engineer an immune response such that you have some immune cells that that attack the tumor. Now, of course, there's the secondary issue. These are hostile tumor microenvironments. They're often hypoxic. They often have a variety of cells that will suppress the endogenous immune response that would also function in suppressing an exogenous immune response from any cellular therapy you would give. But for the majority of patients in the solid tumor world, most of them have immunologic ignorance. So this is the fundamental problem in the solid tumor space. What's gained a lot of traction, as we've heard in lung cancer, melanoma, Hodgkin's lymphoma, renal cell carcinoma, bladder cancer, triple-negative breast cancer, head and neck cancer, so on and so forth, are the anti-PD1 blockade strategies. And so for about, in most of the solid tumor types, 1 in 5 patients, this is what their tumor will look like. And you'll have PD-L1 upregulation and kind of an invasive front at the trenches of T cells that are inhibited from attacking the tumor by PD-L1. And for these patients, an anti-PD-1 based strategy, either as a monotherapy or with the second agent, either a second inhibitor blocking one of the receptors in red, or a TNF superreceptor family agonist, those in green on the earlier slide, will likely maximize an immune response here. And so these patients are often those that will have combinatorial immune checkpoint blockade. For patients who have tumor-infiltrating lymphocytes but no PD-L1 expression, these are likely non-PD-L1 based mechanisms of immune resistance. You have T cells that are present. This is often seen in microsatellite stable colorectal cancer, in which we have not found the right cocktail of immunologics to best unlock an immune response, as opposed to microsatellite unstable colorectal cancers, where there are enough neoantigens that a PD-1 based strategy is sufficient. And so this is an area of active interest, as we see in a lot of our patients who don't fall into this initial category of the immune desert. They do have T cells, but we don't have the right therapies to unlock those cells to attack the tumor. And then finally, with intrinsic induction, these are patients who may have hyperactive ALK kinase signaling or aberrant STAT signaling. These patients will likely require some sort of induction of an immune response, often through a cellular-based strategy or a tyrosine kinase-based strategy, and then potentially with an anti-PD1, PD-L1-based strategy. And this is best seen in melanoma, where BRAF-positive melanoma patients are often given a triplet currently in clinical trials of a BRAF inhibitor, MEK inhibitor, so dual MAP kinase inhibition, plus an anti-PD1 strategy. And so that's really the context of the solid tumor problem. And so in terms of targeting TNF superreceptor family members, if we look at OX40 as one of these members, this was in green at the top, one of the activators. This is expressed during T cell activation and during active engagement of the antigen. So this is a receptor interaction we want to stimulate in terms of our anti-tumor T cells. And also, you can maybe have your cake and eat it too, because these are less likely to result in autoimmune toxicity, since they only work against T cells that are already activated. And that's really one of the keys when talking about combining exogenous cellular therapy, NK cells, CAR T cells, with immune checkpoint blockade, is if this can be done in a way in which you can do this either outside of the body or timed infusions of the checkpoint, such that you're only further activating the anti-tumor specific T cells, avoiding the nascent autoimmune reactive T cells that could result in autoimmune colitis or immune-related hepatitis. These checkpoints are present on a variety of anti-tumor cells, including NK cells, effector cells, and helper cells, and their prevalence depends on the type of tumor we're discussing. And the most important part, and probably the key behind the immunotherapy revolution, has been the durability of responses. When you find the right immunologic agent or cocktail for a patient, the chance that you can generate essentially a vaccine, a long-term effector memory component in that patient to lead to durable remissions, it's something that is really captured imagination and really led to the development of these agents in this field. And so even within expert centers that design their own CAR T cells, there are intricacies in terms of whether to use a 4-1BB agonist or a CD28 in line with the CAR T cell. And interestingly, these are also all monoclonal antibodies that are currently in development to give exogenously. So if you already have a T cell in your body that's ready to attack the tumor, you can use a monoclonal antibody to unlock it. But for most patients, if you're an immunologic desert around your tumor, you will likely need a cellular-based or vaccine-based strategy to maximize an immunologic response. And so the biomarkers really tell the story, and I show this four-part schema. For patients who have high PD1 expression surrounding the tumor, these patients often just require an anti-PD1 antibody, and the response rates are high. For patients who are PD-L1 negative, some of these patients will still respond to an anti-PD1 agent, mainly because the biopsy likely missed the pertinent immune interface between the T cell and the tumor that would have otherwise expressed PD1. But many of these patients will require either a cellular-based or vaccination-based strategy to convert an immunologic desert into a place in which you have a tumor-invasive front of immune cells that are ready to unlock the tumor. And I showed you the melanoma slide, which is the poster child for immune checkpoint blockade in solid tumors. But in terms of the most common solid tumor we treat in clinic, it's metastatic non-small cell lung cancer. And here, patients with high PD-L1 expression do get significant and durable efficacy from anti-PD1 monotherapy. And this just shows that response rates across a variety of histologic subtypes. And as usual, these data look even better in the hematologic malignancies. For example, in Hodgkin's lymphoma, the response rates with anti-PD1 post-auto transplant or post-brentuximab approach 85-87%. Of course, Hodgkin's lymphoma is a bit unique compared to solid tumors because at least nodular sclerosing Hodgkin's lymphoma is driven by an underlying biology of JAK2, PD-L1, PD-L2 amplification. And so that's a different biology than what we see in solid tumors, which is immune-mediated upregulation. And so I want to show this has shown in pretty much every ASCO over the past 3 years, the tumor mutational burden by histology. And essentially, for endogenous immune checkpoint blockade, if you have a T cell already within the tumor, this is essentially a stochastic process. And so if you have either UV-mediated or smoking-mediated damage for these 4 tumor types or 5 tumor types to the furthest right, you have a higher mutational burden per megabase. This increases the chances that there's a cognate T cell within that tumor microenvironment that can be unleashed with immune checkpoint blockade. This is contrary into some of the more pediatric tumors in which there's a single driver mutation. And so in terms of thinking about immune checkpoint blockade, in terms of activating endogenous T cells, the number of neoantigens essentially increases the odds that you'll have a T cell that's ready to be unlocked and attack the tumor. Of course, for patients in which this is not the case, these patients will require vaccination or cell-based strategy. I also want to show that there's some heterogeneity within a given tumor type. And so when you look here at colorectal cancer and here at uterine cancer, these are microsatellite unstable patients. And so even molecular features within a given histology can confer an enhanced ability to respond to immune checkpoint blockade. And so this is the mutational burden story made most famous in colorectal cancer, in which microsatellite unstable patients, either Lynch syndrome or sporadic, meaning they developed a defect in their mismatch repair within their tumor, have several log more mutations per tumor compared to microsatellite stable patients. So these patients, microsatellite unstable patients, often require immune checkpoint blockade, and they'll have high response rate because of this. But the microsatellite stable patients will likely require either a vaccination strategy or cell-based strategy to a unique antigen to unlock an immunologic response, because these tumors are either immune deserts or they're ineffective T cell responses against the tumor. And in melanoma, which is the poster child of the success of immune checkpoint blockade, UV-mediated damage leads to a higher number of mutations. These patients have a higher rate of survival with ipilimumab or anti-CTLA-4-based monotherapy. These numbers look even better when you combine a second immunologic with PD1. And so computationally, what can be done is, in terms of predicting neoantigens, is tumors are sequenced, RNA-seq is performed, and then computational in silico analyses to determine HLA binding are performed for that given patient. And then you can essentially generate a set of candidate peptides. You can peptide load dendritic cells into LE spots to determine biologically which peptide is the peptide that's having an immune response generated to this. And that was done in a set of elegant experiments. This is out of Memorial, in which they essentially looked at the immune peptides and converted them to wild type and looked at differential LE spot and were able to find the candidate peptides from the neoantigens from the tumor that were a correlate of immunologic response, and they could track these serially. And one thing I'd like to point out is you can see these very early in the blood. So they showed as early as day 21, though you probably would have caught it even a week in if they'd looked early enough. And this is essentially the MRD marker in solid tumors for minimal residual disease, and you can actually track this T cell clone against this specific tumor mutation over time. And one of the points in solid tumors is that the response to immune checkpoint blockade is often slower. And so there are a set of radiographic criteria called immune-related response. And because of this, it's often difficult to tell whether a patient's progressing or responding at their first scan. And so there's a second scan that's done. That often takes an additional 2 months. And so the ability for a blood-based biomarker to discern who's responding or not early in their treatment course is important. And then this also leads to the secondary point in which this becomes not only a diagnostic tool, but potentially a therapeutic tool as well. If you can find what the relevant immunologic response is for a tumor, whether you can actually amplify this number of T cells outside the body and reinfuse. And so when we talk about combinations, currently we're talking about combinations of immune checkpoint blockade, and these generally relate to 2 immune checkpoints. But future combinations are really going to be looking at cell-based therapies. And these are currently in clinical trials. There's very little data that's been published, but essentially there are 2 strategies in terms of immune agonism. The first is inline, in which you build the TNF superreceptor family agonist into the CAR T cell or NK product. And so Dan showed the slide on 4-1BB, but there are others, OX40, CD28, co-stimulators that are used as well. This can also be done for cytokines. So this is the armored car T cell analogy, in which you give these T cells that are engineering an IL-12, for example, cartridge that allows these T cells to thrive in a very hostile hypoxic tumor microenvironment that's surrounded by a myriad of immunosuppressive immune cells. And so these are the various constructs, the first generation, second generation, and currently we're on the third generation CARs. Then there's the sequential strategy in which in vivo immune cells, the immunostimulatory antibody can be administered immediately after cellular therapy. And so here you have an activated batch of cells that are infused, and then through activation of various of the receptors in green, the TNF superreceptor family agonists, you can further stimulate those cells and kind of prime them to attack the tumor. And then finally, the ex vivo strategy, in which in the actual bag, after you produce the cellular product, you can give a dose of OX40, 4-1BB, IDO prior to the infusion, with the idea that you can deliver a higher dose and avoid accidentally activating a nascent autoimmune cell that's in the body. And so these are strategies for creating a virtuous cycle within the tumor microenvironment. I know we're running short on time, so I'll kind of go through these quickly. And so essentially when you take a cell therapy-based approach, for example, a CAR T cell approach, these CAR T cells have the same issues that your endogenous T cells do in terms of hostile tumor microenvironment and MDSC suppressing the activity of these cells. And so if you can do PD1 blockade, what will happen is you'll create a virtuous cycle in which there's increased interferon gamma production, which supports these cells, leads to more trafficking of cells, and you convert essentially what was a negative cycle in which PD1 blockade would have shut down the very T cell product you generated into a virtuous cycle in which PD1 blockade leads to further and further engraftment of these CARs within the tumor microenvironment. The other way of handling this is a switch receptor. So rather than giving a monoclonal antibody for PD1, you can actually engineer a CAR T cell in which the PD1 ectodomain actually within the cell becomes a CD28 intracellular signaling domain. Thus, what otherwise would shut down a nascent T cell actually activates it. And so what otherwise would make it weaker makes this T cell stronger. And this is, of course, in parallel to the third generation CAR T cell construct. And this is another way of creating a virtuous cycle with these switch receptors to avoid the hostile tumor microenvironment. And this can be done in NK cells as well in terms of combinatorial strategies. And so in terms of NK cells, whether you want to augment or redirect through a monoclonal antibody-based strategy, combining these with checkpoints, anti-KIR, anti-NKG2A, which binds HLA-E, which is an interesting biology-related placenta that I won't have time to talk about, or various NKG2D ligands. These can be given at the time of your NK infusion to make sure that when these cells reach the ovarian cancer cell or the breast cancer cell, that they're not immediately suppressed. And then finally, cellular engineering approaches in which these bispecific NK cell engagers or CAR approaches can be used to better target these NK cells into the hostile tumor microenvironment, often in combination with checkpoint blockade to make sure these cells thrive. And so really to summarize very briefly, every person's cancer has a unique mechanism of immune invasion. Cellular therapy approaches are very important for patients who have tumors that are essentially the immunologic desert and have immune ignorance, and that they're both T cell and NK cell-based strategies. However, even beyond that, these cells must thrive within a hostile tumor microenvironment within the solid tumor space, and whether this is inline engineering of the cell or sequential therapy, either ex vivo or in vivo, these are all approaches currently being investigated in the clinic, and the idea is essentially the concept of cancer immunotherapy can be broadened across all tumor types given these advanced cellular therapy approaches. And so with that, I think we can go into the panel discussion briefly, potentially.